Good afternoon, learners. I welcome you all to this particular discussion on biosafety issues of genetically modified plants. Now, this particular topic, it belongs to BBYET 143, unit number 15. The topic is applications of plant biotechnology. Now, all of us, in one of my earlier sessions, I discussed about the practical applications of genetically modified plants or the transgenic plants. It has a lot of utility because it has different applications. It has applications in the field of environmental stress tolerance, biotic stress tolerance, in the improvement of nutrition, okay, in the improvement of in the area of increasing the productivity of the crops. So there are different such areas which we already discussed in one of the sessions. But you have to understand that these genetically modified plants or the transgenic plants, they also carry a lot of controversies. They also carry a lot of biosafety issues, because when you are introducing these transgenic plants in the field condition, then you have to actually address a lot of issues, or you have to address a lot of deal with a lot of public opinions or public misconceptions also, you can say. So today we are going to have certain discussions. We will discuss certain points with regard to those controversies, why those controversies, and what are the issues that have been, what are the biosafety issues that we are already addressing in order to popularize the genetically modified crops. Now, to begin with, you have to understand what is meant by biosafety. As it goes in the title, biosafety issues. Biosafety refers to the policies and procedures adopted to ensure environmental safety during the course of development and commercialization of genetically modified organisms. It can be genetically modified microbe, it can be plant, or it can be any other organisms. So you have to follow, or you have to adhere to certain policies and procedures before you can ultimately bring the transgenic crop into the market and commercialize it as a product. This is number one. Number two, efforts to reduce and eliminate the potential risks resulting from biotechnological products. So there are two issues. Number one, whether there is at all any risk with the genetically modified crop, what are those risks? And finally, how will you address those risks, or how will you solve those problems? So these are also included within the biosafety issues. Now, when you talk of biosafety, there are 4 levels: level 1, level 2, level 3, and level 4. Level 1, that is appropriate for the undergraduate and the secondary educational training and teaching laboratories who are working mostly with the characterized strains of viable non-pathogenic microorganisms. So here you don't have any major issue. You don't have any major hazard. So level 1 is perfectly all right. Level 2, clinical, diagnostic, teaching, and other facilities where work is done with broad spectrum of moderate risk agents. So you have to be a little bit more careful here. Specific training is required. How will you handle those microorganisms? How you are going to manage the laboratory conditions? Those things have to be addressed in level 2. Level 3, clinical, diagnostic, teaching, research facilities where work is done with agents having potential for infection and disease having lethal consequences. So here you have to be very, very alert, because you need very specific training. You need specialized personnel to handle these kind of agents, and you also need a very specific, well-designed laboratories to address this kind of work. Lastly, level 4, it is applicable for work with dangerous and exotic agents. So this is very, very alarming. You need a very, very strict training and supervision is required, and specifically designed laboratories are required with stringent safety condition. You have to be very careful so that there is no contamination, so that from the laboratory there is no contamination. Like you are studying with any virus, okay, and that virus suddenly it comes out of the laboratories. We have seen what could be the consequence of that. In the last 2 or 2.5 years, we have been seeing that. So you have to be very, very cautious in handling this kind of organisms, virus, bacteria, whatever. Now, in order to address these issues, the Department of Biotechnology, Government of India, in collaboration with the Ministry of Environment and Forests, they are the nodal agency and they have framed certain guidelines regarding how will you work with these genetically modified organisms, plants or microbes. Today I will be mostly focusing on the plants because my area of specialization is of course plants. So there are actually 5 such hierarchical tiers, you can say. To begin with, the recombinant DNA advisory committee. Whenever you are working with any recombinant DNA molecule, this committee or this agency will take care of that, and they will recommend certain safety regulations for research and applications. Number 2, institutional biosafety committee. That means in the institution where a student or the scientist or the academician is working, they should have an institutional biosafety committee, and there should be one DBT nominee, that is nominee from Department of Biotechnology, who will look after what kind of research activities are going on or what kind of work is going on. Okay, so that is the main task of the institutional biosafety committee. So to note and approve the recombinant DNA work, to ensure adherence of the recombinant DNA safety guidelines, to prepare emergency plan according to guidelines, and to seek the recommendations and approval of RCGM. I am coming to that. RCGM stands for Review Committee on Genetic Manipulation. So the next tier that we have is the Review Committee on Genetic Manipulation. Now this committee has several activities, or they have several responsibilities. Bring out manuals of guidelines specifying regulatory procedures on genetically modified organisms in research, use and applications including industry. Review all the work which are going on in rDNA projects, recombinant DNA work, including risk category and controlled field experimentation. Lay down procedures for restriction or prohibition, production, sale, import and use of genetically modified organisms. Permit experiments with risk factors. Authorize field experiments in 20 acres in multi-locations in one crop season. Generate relevant data on transgenic materials, and undertake visits of sites of experimental facilities periodically. So in short, what I mean to say is, I just summarize what this committee is actually looking for. Number 1, if you are working with a genetically modified plant or an organism, then ultimately if you are working with a transgenic plant, you have to bring the plant outside someday. Okay, you have to undertake field trials, whether your transgenic plant is performing well in different agroclimatic conditions. So you have to undergo field trials. Now this committee will actually take a note of that. Okay, whether these field trials are being conducted, how many locations you are carrying out these field trials, what is the area of land that you are using, how you are conducting the field trials. Okay, and if you are trying to commercialize your product, that is genetically modified product, what are the protocols that you are following? Okay, if you want to export your transgenic product or genetically modified product, how you are doing that? So all these are actually taken care of by this committee. The fourth committee that we have is the Genetic Engineering Approval Committee, or GEAC. This is also a very important committee which is under the control of Ministry of Environment and Forests. They are also performing several tasks like permit the use of genetically modified organisms and products, adopt procedures for restriction or prohibition of sale and import, whether your product is safe, whether it carries any environmental risk. Okay, if yes, then this committee will actually stop that. It will not allow you to commercialize your product, okay, or to bring it into the market for the sake of benefit of the mankind. Authorize large-scale production and release of genetically modified organisms and products. Authorize agencies or persons to have powers to take punitive action under Environmental Protection Agency. Because if you violate, if you don't follow the stringent rules and regulation before commercialization of your product, then this committee can take stringent action against you. So these kind of checkpoints are there always. Okay. And lastly, we have the last two committees that we have: State Biotechnology Coordination Committee. In each state, there is a State Biotechnology Coordination Committee, which will be headed by the Chief Secretary, where the research and applications are contemplated. And finally, at the district level also, you have the district level committee, which is headed by the district collector as an authoritative unit to monitor the safety regulations. So you see that there are a lot of checkpoints. So there is nothing to worry about with because we have a lot of misconceptions with the transgenic plants. So I will try to address some of those also, but to start with, we have different agencies who are actually looking after the safety issues. Now, regarding the guidelines for research in genetically modified plants, again, DBT has categorized this kind of, research into three categories. Number one, category one, there is absolutely no problem in category one because it involves routine cloning of defined genes, mostly E. coli or any other hosts which are absolutely safe to humans. So if you find any laboratory who are working with this kind of agents, then you have to understand that they will come under category 1. So there is nothing to worry about. Category 2, that is experiments carried out in laboratories and greenhouse or net house. Supposing I generate a transgenic plant. So my first task will be to grow it in the tissue culture condition and then bring it out in the greenhouse condition or the net house conditions, and I grow there till it matures or till it grows, okay, till it gives the first generation seed. So that comes under category 2. And category 3, you have to be very careful because now after that you have grown the transgenic plants in the greenhouse, you want to bring it in the field condition. Now when you bring it in the field condition, you have to address a lot of issues because there are other wild type plants, there are normal plants, traditional varieties. So you have to make sure that the traditional varieties, they don't get contaminated because you have introduced your transgenic plant into the natural environment. So category 3, if you are doing this kind of work, then it will come under the category of category 3, and then you have to again follow those stringent rules and regulations that have been embodied in the DBT guidelines. That is, you have the different committees and your work will be under the close inspection or examination of the committees that I discussed of the DBT. So those kind of work will come under category 3. So these are the guidelines that need to be followed. Next is what is meant by containment in case of biosafety. Containment means you limit the spread of genetically modified organism and its genetic material to the environment. That means you don't want to bring the genetically modified material into the environment. What will you do? Then you have to follow a containment. Containment means you have to confine it. How can we confine? There are two ways of confinement. One is physical containment. Physical containment means you don't allow the transgenic plants to come directly into the environment. That means you confine it within your greenhouse condition. You establish a greenhouse in your institution where you are working. It can be a university or it can be any research institute. You construct a greenhouse and you confine your transgenic materials to the greenhouse condition. Okay. And the second one is the biological containment. Biological containment means you genetically modify the plant in such a way that there is no chance of contamination. There is no chance of pollination, because we know that it is because of the pollination or pollen escape that a surrounding plant can be contaminated. So in that case, there are different techniques in plant biotechnology. There is a way of making male sterile lines, or there is a way of there is a technique called chloroplast transformation, because chloroplast, we all know, it is maternally inherited. So there is no chance of pollen escape if you do chloroplast transformation, unlike nuclear transformation. So these are some of the techniques which the scientists have actually adapted so as to not allow pollen escape from the transgenic plant to the wild type plant and thereby contaminating the wild type plant, because this is one of the serious issues of the transgenic crops or the genetically modified crops. Now, I will give some of the examples which had happened in the past, for which we are very much confused whether we will accept the genetically modified crops, whether we will accept the transgenic product or not, whether it will be safe for us, whether it will not cause any adverse effect, either to our health or to the environment. So these are some of the background or some of the incidents which had happened in the past, which actually created some sort of confusion in our mind. Now, one of them is the starling corn incident. What happened? Cry9C, cry stands for crystalline toxin. In one of my earlier session, I talked on Bt transgenics, Bacillus thuringiensis, which actually leads to the insect resistance. So in order to generate insect resistance, a cry gene called cry9C gene, it was actually cloned in corn, that is maize. Okay, and the plants were found to be insect resistant, but later on it was found that the people consuming this transgenic maize, they were acquiring some sort of allergenicity. So there was a suspected allergenic reaction in certain human populations. So in that way, the starling corn, it was actually abandoned because it was not accepted because it created some sort of allergenicity. A similar situation was there, that is case 2, when you actually clone the albumin protein from Brazil nut and you clone it into soybean. Now, why this was done? In order to enhance certain sulfur containing amino acids, in order to biofortify soybean. But it was again seen that when these transgenic soybean were consumed, it was found to be allergenic. So this created a lot of, it created a lot of confusion, and people were very much, they were very much unhappy with the product, and they were very much confused with the product, and it was also abandoned. So this was second case. Third case, it might happen that sometimes a non-immunogenic protein, it becomes immunogenic when expressed in another species. You know that the protein is not causing any major harm. But when you introduce it in certain in a recipient plant, then suddenly if you consume that, you found that there is an allergenicity. So some sort of reactions have happened that actually cause this allergenicity in certain population. Even it was first tried in an animal trial was made, and it was found that there was an immunogenic response. So these were some of the issues. So to be precise, number one, it is allergenicity, induction of the allergenicity. Number two, antibiotic resistance. This is another, because when you are transforming any plant, you are making a genetically modified plant, then we have to actually go for the selection. And therefore, there is an antibiotic resistance gene which is present in your plasmid vector, and that is actually transformed into the plant, and you select the plant under antibiotic selection. Now, this also has created a lot of confusion, because it is believed that if you are consuming some substances which has antibiotic resistant trait, then you are actually creating a situation where the microbes, they might acquire the antibiotic resistant trait due to the horizontal gene transfer, and therefore it might be very difficult for you. If you get infected with certain microbe, then you might get it might be very difficult to control that disease using antibiotics, because the microbes, even if they are pathogenic at all, then they have acquired the antibiotic resistant trait, and therefore it will be very hard to eradicate them. So this is another issue. But scientists have actually seen from various studies that the horizontal gene transfer from the plant material to the gut is extremely unlikely. It's very, very unlikely that from the plant it will go and enter into your system, okay? Because there are different intestinal barriers. We have an alkaline pH. In the stomach, it is acidic pH. So under such stringent pH conditions, it is highly expected that these antibiotic properties will be lost. Moreover, if you eat cooked food, for instance, if you eat staple food like rice and wheat, which are cooked food, so the antibiotic property, resistant property, antibiotic is already destroyed, by high temperature or heat, or under such acidic conditions. So there is no way that this antibiotic resistance is going to affect you in any way. Second is certain antibiotic resistance genes, they are ubiquitous. Ubiquitous means they are present in nature as such. It is present in any organism. It is present in humans. It is present in any other animal, even in plant. So it does not affect much. Something which is present universally, it does not affect much. So these are the 2 points which one should be you should be quite happy to know that it does not cause any major issues. Another point, that whether it is an environmental hazard, because as I said that when you are introducing a transgenic plant in a field condition, then there is a chance that because of pollination, the surrounding plants which are non-transgenic, they might get affected, and this is called the genetic erosion, because you are actually losing the traditional varieties which are already present in the field, or the because those traditional varieties, they might get contaminated because of the pollination. But I already mentioned that we can address the pollination issue, because if we go for chloroplast transformation, then there is no such pollination issue. And scientists are actually doing that. They are trying to create male sterile lines, or they are trying to undergo chloroplast transformation so that there is no risk. Another point, that is creation of the superweeds. Superweeds means if the GM crops, they hybridize with certain weedy relatives. Supposing in the agricultural field we also have the weeds, and if they hybridize with the weedy relatives, then it might lead to the creation of the superweeds. And in order to eliminate the superweeds, it might be that the farmers have to use more and more, this kind of pesticides, or high doses of herbicides or pesticides, mostly the herbicides, not pesticides, because pesticides are meant mostly for the insect pests, the herbicides, and so that will be an indirect way of contaminating the environment. So this is another issue which people actually are concerned with. So that is actually the ecological hazard or the environmental hazard. Now, the point is that this gene flow, that when I say that the local varieties or the non-transgenic varieties, they will get contaminated with the transgenic varieties, here scientists have actually addressed the issue through 3 ways. Number 1 is creation of an isolation zone. Now, what is meant by isolation zone? That means you try to confine, even if you bring it to the field condition, you try to confine the transgenic plants within an area and not allow the normal plants or the non-transgenic plants to grow nearby. Because if you separate a distance of 8 meters between the non-transgenic plants and transgenic plants, then there is very little chance of pollination, and there is very little chance of getting contamination of the non-transgenic plant because of the pollination. So the 8-meter distance can be maintained. Okay. Number 2 is trap crop. Trap crop means non-transgenic varieties of the same crop planted adjacent to the transgenic plot can clean up pollinators of transgenic pollen, preventing gene flow. Third point, male sterility I already told, and chloroplast transformation I already discussed, because the crops will not produce pollen, or they will produce inactive pollens. So there will be no pollination. And chloroplast transformation, because chloroplast is maternally inherited, so there is no chance of pollen escape, no pollination, and therefore there is no chance of contamination. So these are some of the techniques which the scientists are following, okay, so that you don't actually contaminate the environment. Okay, so it is environmentally and ecologically safe. There is no hazard. Another point, that is loss of biodiversity. That is whether the traditional varieties will be lost, whether there will be a genetic erosion, because you are replacing the, you are not replacing, rather you are introducing the transgenic plants into the field condition. So this is whether it is causing a loss of biodiversity. The answer will be again no, because if you maintain that isolation zone, or if you try to confine the transgenic plants within a restricted area, then there is no chance that the non-transgenic plants or the wild type plants will get affected. Another issue is non-target effects. That means whether the transgenic plant, like if you generate an insect resistant plant. Now insect, when I am trying for BT transgenic, then I already discussed in one of my sessions earlier that the BT transgenics are only meant for insects, and that also a specific group of insects. So there is no chance that if you introduce BT transgenics, BT brinjal or BT cotton into the field condition or in the environment, then all species of insects or all animals will get affected. No, only the insects, and that also a specific group of insects. So it's you have to generate a transgenic, you have to understand the gene and its function so that it does not create any non-target effect. It should be precisely targeted to the specific microorganism that you are wanting. So that has to be very carefully studied, and if that is being done with care, I don't think that there is any such concern or issue of introducing the transgenics into the environment. Now, in order to address these issues, the Cartagena Protocol on Biosafety, it was actually framed. It was finalized in February 1999 at Cartagena in Colombia, and it was adopted a year later on January 29, 2000 in Montreal, Canada. And it entered into force on 11 September 2003, and including India, 147 countries, they have ratified this protocol as of May 2008. So India has also signed this protocol, and because India is also one of the countries, I will show some of the mega countries which are actually involved in generating biotech crops, and India is also one of them. So how will you handle this genetically modified crops? How will you come up with a product? How will you commercialize the product? Okay, how will you export that product? So all these guidelines, they are actually embodied in this Cartagena Protocol on Biosafety. So there are a lot of rules and regulations, but I will focus only on the main ones. Number one, supposing you have generated a product, transgenic product or genetically modified product, and you want to let's say export it to some other country. So it will involve cross-border movement of the transgenic germplasm. Now how will you do it? Now in this case, the review committee on genetic manipulation will issue an import certificate after inspecting the documents related to the safety of the material and national need. The importer may import the material accompanied with a phytosanitary certificate. What is phytosanitary certificate? Phytosanitary certificate is a certificate which actually will be issued, and it will show that this product is free from any pathogen, this product is absolutely free for the environment, this product is absolutely safe for consumption, and all other details. Because when you are bringing one item from another country, then you have to ensure that it does not carry any other propagules or any other germs or any other pathogens along with it. Okay, otherwise it might create an epidemic condition. Okay, so that has to be, or even maybe, who knows, even a pandemic situation. So you have to be very careful about that, and therefore this the importance or implications of this phytosanitary certificate. So this is issued by the authority of the country of export. The import should be routed through the director NBPGR. NBPGR stands for National Bureau of Plant Genetic Resources, on the basis of import permit issued by the Review Committee on Genetic Manipulation. So this is with regard to how do you export and import any product which is genetically modified. So the import certificate will be cancelled if NBPGR is not provided the phytosanitary certificate. So if that is lacking, if that certificate is not issued, then it cannot be, it cannot undergo the transboundary movement or the cross-border movement. Part of the seed material will be kept at NBPGR in the presence of the importer for legal dispute. So a sample, a small fraction or a small aliquot of the sample will also be kept with the NBPGR, because if there is any kind of confusion later, or if there is any legal case later, then that can be addressed by the NBPGR. So this is with regard to the transboundary movement of the transgenic germplasm. Next we have Biosafety Clearing House. Now it contains information on national laws, regulation and guidelines for implementing the Cartagena Protocol. It contains information required under Advanced Informed Agreement procedure. Now what is this Advanced Informed Agreement? In short it is called as AIA. That means if I want to import any transgenic product from any foreign country, then there should be a mutual consent. I mean, that country should agree, okay, I should also agree whether I accept that particular product. So there should be a mutual consent or a mutual agreement between the two, okay, regarding the procurement of the germplasm. Okay, and that consent should be there, then only I can go for this kind of transboundary movement. So that is clearly spelled out, or that rule is clearly spelled out in this, under this biosafety clearinghouse. Summaries of risk assessment and environmental reviews, as well as other scientific, environmental and legal information. So these are actually, so this is an internet based system, and you have this particular link, that is http://bch.biodiv.org. So this is an internet based system, and you have all these rules and regulation, okay, regarding the BCH in this particular link. You can find in this link. Next, the point that we have, that is Codex Alimentarius Commission. This is also a very, very important what should I say, rule with regard to the commercialization of the product, because the consumer who actually consumes a transgenic product should know that I am consuming the genetically modified product. Okay, when I purchase, okay, supposing I go to a mall and I find that there is a genetically modified crop, but I should know that it is a genetically modified crop. So there should be a specific label. It should be properly labeled that this is a GM product, and the consumer knows that he or she is purchasing that GM product and consuming that. So this is actually, so there should be a label. There should be a proper mention. So Commission is responsible for labeling biotic foods to allow the consumer to make an informed choice. So whether I prefer, or whether I have a choice, or whether I really choose that particular product, that should be well known to me. So that is spelled in CSC, or the Codex Alimentarius Commission. Next, we have the substantial equivalence. Substantial equivalence actually says that the genetically modified product that you have made should be absolutely identical to the normal plant or the wild type plant, with the exception that it has one value added trait. What is meant by that? Supposing you want to generate golden rice. Now, what is golden rice basically? Essentially, it is rice which is having which is rich in carotenoids, which is rich in beta carotene. So all the properties of rice should be there in golden rice. It should not differ in terms of productivity, okay, the seed setting, yield, okay, its response to the environment. So all the properties should be there, with the exception that it has one value added trait, which is that it is rich in carotenoids. So that you have to show whenever you are generating such a transgenic product. You have to show that it is exactly identical to the normal plant, with the exception that it has one value added trait. So that is actually spelled in substantial equivalence. And mind that whenever you are going for commercialization of product or marketing of a product, okay, you have to address this substantial equivalence issue. So these are the first part of my discussion actually was focused on biosafety issues. Now the second part of my discussion, I will talk on the problems, controversies and the misconceptions which are there, which are highly prevalent in the society, because till now it's our what should I say, it's really a pity that we could not commercialize, excepting maybe BT cotton or maybe BT brinjal, not even BT brinjal, BT cotton. We could not bring any transgenic products into the market. Okay, so why? The question is why? Because we have a lot of misconceptions, we have a lot of confusions in our mind. So I am going to discuss on those, some of the controversies or some of the misconceptions as well. Now these 2 points I have already I think mentioned. One is pollen escape and another is whether any antibiotic resistance gene causes any hazard. Now it actually causes no hazard, number one, and number two, even if I take it into consideration that presence of antibiotic resistance gene is not safe, then also the scientists have addressed that issue by generating antibiotic resistant, antibiotic free or marker free transgenic plants. Because the scientists have actually cloned a particular gene called managene, which encodes for phosphomannose isomerase, and that phosphomannose isomerase is capable of converting mannose to mannose 6 phosphate followed by fructose 6 phosphate, which enters into glycolysis. So if you grow the transgenic plants which is having managene in presence of mannose, then the transgenic plants can grow happily because they will be able to convert the mannose to mannose 6 phosphate and then fructose 6 phosphate. But the non-transgenic plants or the wild type plants will be unable to utilize that because managene is not there, and therefore in presence of mannose the plants will die. So in this case there is no antibiotic resistance gene, but even then you are undergoing the selection procedure. So scientists have actually done that in order to minimize the level of controversy surrounding the transgenic plants. Now I will show some public misconceptions one by one. Now the first point is, the general perception is that the plant biotechnologists, when you are engineering something, you are actually assuming the role of God. I mean, there are certain religious restrictions. What are those religious restrictions? That you are playing with nature. We as humans, we don't have the right to play with nature. We are actually tinkerers. We are tinkering with the nature. We are manipulating the nature. And we as a human have no right to manipulate the nature. This is absolutely, it comes from a religious faith. But the thing is that, irony is that gene transfer process, pollination, it occurs in nature. Can we stop pollination? Wind pollination. The air, it actually, the pollens actually, they travel because of the air movement. Anemophily, we have one particular terminology in botany, that is anemophilous plants, where the pollination occurs by wind. So can we stop that? So pollination is actually a way by which gene transfer is occurring in nature. So nature is doing that. We, the humans, we are not the only one who are doing that. Nature is doing that herself. So can we stop that, or do we ever try to stop that? Does that carry no chance of cross-contamination? This is point number one. So we don't play the major role. It is the nature which is playing the major role, number one. Number two, farmers for the last 10,000 years, they have been undergoing selfing or hybridization program. In plant breeding, we actually cross two varieties. But see, we don't have any controversy with plant breeding or hybridization program because you cross two varieties. Here also you are exchanging the genes. A superior variety having a superior gene, you are incorporating it into another variety through crossing. But only with the development of a transgenic or genetically modified plant, the entire moral stance changed overnight, and we have a lot of disputes, or we have a lot of controversies. When you have to understand that this transgenic is more precise, whereas hybridization program, this is more, it is more non-specific, because even along with the target gene, a non-specific gene may also be transferred because of selfing. But see, we are more concerned with the transgenic, and we are not that much concerned with the breeding program or the hybridization program. So this is another misconception that we have in our society. So that particular slide actually shows that the plant biotechnology or transgenic is more specific because you are targeting one particular gene, whereas in case of traditional plant breeding, it can be non-specific. Now, I already said that we have a lot of we have a biotech mega countries. There are several countries, as you can see, of which India, China, Pakistan, even Australia, United States is of course there. So there are several countries who are gradually trying to, bring the transgenic crops to the field condition. We are trying to popularize it. But the main thing that I want to focus is it is our public mind or public perception that needs to be changed. Otherwise, we cannot bring these products into the market. We are doing a lot of research. We are doing very good. But ultimately, when it comes from the point of view of market or commercialization, we are a big zero. And why? Because we have a lot of misconceptions. That has to be, everything has to be scientifically judged. Now, another question, is it a solution to world hunger? Is it actually doing good for the society? This is another question. And means for equal sharing of technological benefits. Now, what do you mean by equal sharing of technological benefits? That means whether it is actually doing good for the poor people, whether it is actually doing good for the farmers, whether it is actually trying or whether it is actually capable of mitigating world hunger. These are the questions. That means you are doing the technology, but whether the technology is shared equitably among the common masses, among the common people, among the poor people, among the farmers. The general opinion is that the multinational seed companies are selling the seeds at a very high price. And the farmers, poor farmers, they are incapable of purchasing the seeds because multinational companies are selling the seeds, the transgenic seeds. They are trying to keep it as their monopoly. And the farmers, whenever they approach these multinational companies, they have to pay very high. So the common perception is that it is not reaching the common mass. It is not reaching the poor farmers. So what to do? So the need for profit has attracted the interest of private sectors and multinational companies to defend their investment in crop biotechnology with patent and intellectual property right. Because a scientist, whenever a scientist is trying to has identified a particular gene or has come up with a technology, the scientist actually tries to patent it. Because we all know about the basmati rice case. We all know about the turmeric case, the neem case, how it has been biopirated or how it has undergone biopiracy. It has been stolen by some other countries when it was our indigenous resource. Now, whether the scientists should go for patent, whether the multinational companies should go for patent, that is also a very that is also a question which is of great controversy, because sometimes you will say yes, they should patent, otherwise any country might take the product. Again you will say that no, they should not patent, because if they patent then it will not reach the common mass. So you see, it does not have a fixed answer. Okay, so whether this means that the multinational companies or the scientists, they should stop implementing the patent or the intellectual property right? If yes, then how will you stop biopiracy? So you see, the things are not very easy to explain or understand, okay, or to justify. Now, problems like poverty, unequal distribution of food, overpopulation, ownership of genes, and excessive commercialization of GM crops by the multinational companies. So the real root of the problem is socio-economic and not mere the transgenic technology itself. You should not blame the transgenic technology. Rather, it's actually a more social issue or a socio-economic issue that has to be understood. Now, another issue that is also very, I would say, a public concern. There is a misconception in the society that if you clone a bacterial gene into a plant, then actually the plant will turn pathogenic because you are introducing bacteria into a plant. It's absolutely a misconception. If you clone an insulin gene in a plant in order to generate an edible vaccine, then it's not that the plant will become a non-veg item. The plant will remain a plant. Plant will remain a veg item. It will not become a non-veg item because you have introduced insulin gene in order to generate edible vaccine. But you see, we have a kind of public perception that you are introducing insulin gene in plant and the plant becomes a non-veg item. It's not that. Is it ethical to slaughter pigs or mouse for isolation of insulin? You kill the animals to isolate insulin from them. Is it equally ethical? The answer is no. Then how will you generate insulin in a shorter time? It can be that you generate edible vaccines. You consume insulin along with the plant system. Now, another point that also needs to be seen, that all these factors together, they have cumulatively led to the removal of GM products from restaurants, schools, and supermarkets. You don't find anywhere any GM product being sold. But the irony is that the markets, they prefer to sell tobacco, alcohol, fatty acid food, which creates obesity. And see, we don't raise any question against them. Rather, we are happy to purchase them. But only when it comes to the point of genetically modified crop, we shout. This unjustified situation has to be understood. It has to be properly judged scientifically. Now, the socio-economic impact is a technology came in early 2000, which is known as seed terminated technology, which of course is a matter of huge dispute. Because what is this terminated technology? There were some multinational companies who actually generated plants, or they created transgenic plants in such a way that the plant will only produce fertile seeds for only one generation. Okay, but if you sow those seeds, the second generation plant will not be raised. So what is the consequence? The farmers will approach the multinational company every time to purchase new lot of seed. They will grow it, okay, they will generate the second generation seed, but when they sow those seeds, okay, they are not getting any plant. So this kind of technology was developed. Of course, this technology has been totally banned, and it created a lot of furor, or it created a lot of dispute in the environment. And of course, this kind of technology should be stopped. But the other side of the coin is that just because some multinational companies are doing this kind of misconduct does not mean that the technology is wrong. So you have to understand that. Of course, there will be a certain school of people, or certain school who will be doing this kind of misconduct, but you have to take proper steps to stop them. But don't stop the technology, because currently we don't have any alternative strategy or technology to increase the food productivity in order to mitigate the global hunger. Okay. So the fundamental concerns, the religious faiths condemn transgenic by saying it's sinful to meddle with nature and it goes against God's wish. I told you that we have a misconception, especially the religious faith. It says that we are tampering the nature. Now the question comes then, where does divine responsibility end and man's responsibility begin? So where from do we start as a human? What is our responsibility as a human in order to save our nature, save our food, okay, to meet the acute food crisis? We have to do something. How do we do that? So transgenic and engineered sound more sinister than eugenic and domesticated. Another issue, that has life been reduced to a series of gene products only? You are generating products. So everything becomes commodified. This is another question which comes in our mind. Commodification of nature and evolution is being engineered. This is another kind. So these actually comes from different religious faith or religious schools. So in the end, I'm almost towards the end. Now, like any other technology, controversy surrounding GM crops is no exception in a heterogeneous society. If you do any good, it will not only have positive side, it will also have some negative side. I mean, it will always invite some sort of controversies. So similar is the situation with this transgenic or GM technology. Several concerns need to be addressed on different issues like food safety, conflict between plant variety protection and IPR. That is, I already said, whether you should go for IPR, intellectual property right, patent, whether you should go for it or not. Okay. Finally, successful product development needs extensive field trials and public understanding. You have to undertake extensive field trial to show that your product is absolutely safe. Okay. Animal trial also should be done, that it is absolutely safe. It is not creating any adverse effects or allergenicity. It is absolutely safe for the environment. Okay, so these kind of trials should be of course made, and of course we have the different, I told you, the committees of DBT are there who will be taking care of whether all these kind of experimentations have been properly conducted. Scientists play a sensible role by convincing the policymakers to expand further the application of genetic engineering in modern agriculture and human welfare. Current public opinion, to be very frank, is based more on emotions rather than scientific judgment. We are judging this emotionally rather than scientifically. So that should not be the case. We should come up with a rational mind. Of course, it has some negative sides, but we should come up with a rational thinking and rational opinion and try to at least allow the conduction of the experimentations and the field trials that the scientists are eagerly looking for. Because if you stop them, if you stop the scientists from bringing this product to the market, then it will be in the embryo stage only. It will never germinate. So that is the thing. Whenever change has occurred in a society, there has initially been certain ripples. If you throw a stone in a pond, there will be a little bit of ripples initially, but then again it will stop. So probably with that hope that someday this technology will become more and more popular, let's hope for that, because otherwise we are not able to solve this human hunger. You know how many, what the percentage, huge percentage of population who are not getting proper food, okay, because of the food scarcity. So we should think of some technology or some way by which we can solve that particular problem. So for the full potential of transgenics to be realized, there should be a proper communication. Scientists alone cannot do anything. There should be an unison, or there should be a proper communication between the different groups: scientists, politicians, journalists, and common people. Journalists have a very important role, because if you write something which is not scientific in a media, then the people will believe that. So all of these people, they should come under one umbrella. They should be united, and then then only we can make the mass understand. So if you compare the benefits and risks, you find that the benefits far outweigh the risks. Benefits are more rather than the risk. So in the current situation, this technology is the only solution to boost agricultural production, to boost up agricultural production, protect crop plants, and alleviate hunger of ever increasing population. Because if you see very scientifically, you don't have any other alternative technology which can generate, because plant breeding or hybridization, it takes a lot of time, because you have to go on selfing, you have to go on crossing, go for the back crossing, okay, and it takes several years to generate one particular product. Of course, in India, most of the products that we see in the market, the hybrid varieties that you see in the market, those are actually, they have come due to plant breeding or selfing or hybridization. But it has taken a lot of time. This technology, in that way, it takes a much shorter time. So that's why we need to appreciate it, number one. And therefore, further research should be conducted, or it should be allowed to conduct. If don't stop the scientists, you should allow the scientists to carry on this kind of work, because there is no alternate promising technology that can mitigate world hunger. So with that particular, what should I say, with that orientation or with that thought in mind, let's accept this technology, let's address all the biosafety issues, let's not go for the misconceptions. Even if there are controversies, judge those with a very scientific mind, and then only we can appreciate this technology and we can get the best benefit out of this technology. So I thank you all for your nice listening, and you will also find in Unit 15, as I said, BBYET 143, but I have discussed the topic in a much greater details. In your unit, you may be finding it in a very discussed in a short way, in a brief way, but I have elaborated it so as to give you a more elaborate idea. And I also would ask you or encourage you to think, okay, in this line. Thank you all for your nice listening. Have a very happy learning.
Well, viewers, School of Extension and Development Studies has launched PG Diploma in Extension Development Studies, Masters in Extension and Development Studies, and Certificate in Extension and Development Studies. In addition to this, there are two more